And then before you rush off, I'll remind you again that we'll take a quick group photo. So now our next speaker, Prof. Chipman, has talked to me about polarization, imaging, and all sorts of great things like that. Thanks, Rob.
Thank you very much, and I want to welcome you all here to this College of Optical Sciences. I think this is a really great meeting, and I'm glad to meet lots of you and participate in this. Polarization is a very large subject that affects all areas of optics. I'd like to focus in particular here on remote sensing, so I can tell you a lot in detail about one area rather than a little bit about a lot of different areas. So I want to talk about polarized light and then polarimetry, which is the measurement of polarization properties, and then give you some examples of the polarization in the natural environment. So like you could ask yourself, how polarized is this scene right here, with us? And we have no polarization sensitivity in our vision, unlike bees and octopuses and salmon and many other animals that can see polarization and use it, as part of their survival strategy. But humans and other mammals have lost the ability to sense polarization. And then I want to show you an imaging polarimeter I've been working on for 12 years. We've built several generations of it that we use to measure these natural polarization signatures. And now this has morphed into a program for a space-based polarimeter that's going to fly in a few years. And so my laboratory is participating with the Jet Propulsion Lab on this instrument, Maya, that's going to study aerosols in metropolitan areas for health questions. So, again, I just want to welcome you here, and I run the polarization laboratory, and we're focused on polarization measurements and polarization in optical design, which I'm not going to talk about today, but I would love to talk to any of you who are interested, on the side about how we incorporate polarization in ray tracing and analysis. So, what is light? And I have a really hard time telling my mother, what is light, because she doesn't really understand what an electric field is or a magnetic field. So, I have a hard time explaining it to her, but I hope that you are all comfortable with those concepts, because light is a transverse electromagnetic wave. We have an electric field shown here in blue and a magnetic field shown in red, and they oscillate in phase with each other. And for linearly polarized light, this electric field just oscillates in a particular plane. And by convention, we use the electric field to define the orientation of the polarization. So if I say horizontally polarized light, the electric field is in this plane, for example. And for monochromatic light, well, that's light of a single frequency. And so the electric and magnetic fields are purely sinusoidal. And we have laser light, which is nearly monochromatic, but light in nature isn't very monochromatic. Here we show examples of the electric and magnetic fields for linearly polarized light at two different orientations. We also have elliptically and circularly polarized light. Here's a picture of the electric field going around in circularly polarized light. And so for monochromatic light, we can define a polarization ellipse that describes that monochromatic polarization state, where the light has some amplitude in the x direction and some amplitude in the y direction and phase in the x direction and phase in the y direction. And we often draw a polarization ellipse to describe the polarization state. But looking at this ellipse, it's a little misleading, because what this ellipse really means is an electric field that's going around like this. So this diagram is a shorthand for this. Okay? The electric field isn't doing this, it's doing this. So these are representations of the polarization for monochromatic light, but in nature we have very little monochromatic light. The light is polychromatic, and we can express it as the sum of monochromatic plane waves. And then we have general wave fronts, which aren't plane waves at all, but are arbitrary shapes. We like to make spherical waves in optical systems because we can focus them very nicely. But we need to characterize the polarization of these general light fields. And just to get a quick idea about polychromatic light, here we have 4 different monochromatic waves, 4 different frequencies, a high frequency purple, a blue one, a green one, a red one. And if we add all of these electric fields together, what we're going to observe for the electric field is something irregular like this, where it's hard to define the period because there's 4 different frequencies in this. And so if we actually look at the light that surrounds us, we will see some much more irregular shape to that electric field based on all of the different wavelengths that are present in their amplitudes and phases. And just as we have this irregular oscillation in polychromatic light, we also have an irregular oscillation in the polarization. And so to describe polychromatic light, instead of going back and talking about the amplitudes and phases, as we do here, well, every wavelength would have its own amplitude and phase, and I would need a very long, very detailed description if I wanted to describe my polychromatic light in this form. So shorthand was developed back in the 1870s for describing polarization, long before they knew about monochromatic light, which are called the Stokes parameters. And the Stokes parameters are defined in terms of measurements that we could make with ideal polarizers. And so there's 4 Stokes parameters. We can call them S0, S1, S2, or S3, or they're also called I, Q, U, and V. And the first Stokes parameter is just the intensity of the light. If I measure with a radiometer, I would get a certain intensity. That would be the same number I would get if I measured through an ideal horizontal polarizer and an ideal vertical polarizer. That would give me 2 components that would add to the total intensity of the light. And then if I take that horizontal and that vertical measurement and I subtract them, that will tell me if there's a tendency for more horizontal polarized light or more vertically polarized light. Similarly, I can subtract measurements at 45 and 135 degrees and get an S2 parameter. And I can subtract a measurement with a right circular polarizer and a left circular polarizer and get an S3 measurement. And now with these 3 numbers, S1, S2, and S3, I can reproduce that polarization ellipse. Okay? This is enough information to describe the polarization ellipse. But we have a general method here which is now very applicable to polychromatic light. And it's very simple and straightforward to measure. And we don't usually do the measurement with these 6 particular polarizers. There's many different ways we can do the measurement to get the same 4 numbers. This is just the definition. It's the simplest way to think about what these parameters are. And so what I want to do for much of the rest of the talk is show you some of these Stokes parameter images and talk about how we can understand them, how we can use them, and give you some sense of the polarization in the environment. So one of the first things I want to know is my light polarized or unpolarized? Like sunlight is unpolarized. And so we use a parameter called the degree of polarization to describe, is the light completely polarized, is the light unpolarized? If the light's completely polarized, then there is some polarizer that will transmit the entire beam. All of the wavelengths, all of the energy will be transmitted. And so I might have many different wavelengths in here, purple, blue, green, red, but if they're all oscillating in the same plane, then my entire beam is polarized in that direction. But if every wavelength was polarized in some random orientation, and I had hundreds of random polarizations, and they all added together, then my electric field would look very random like this. So over here I have something that's in one plane. Here I have some small variation of the polarization, increasing randomness in the polarization, and then I have completely random. And so my degree of linear polarization when it's 0, well, any polarizer will pass half of this light. Okay? When I get over to here, there's a polarizer that will pass all of one polarization state, and there must be another polarizer that would block the entire beam. and then we have these intermediate states. And the light that arrives directly from the sun is basically nearly unpolarized. But after it scatters from the sky, it can become partially polarized, or after it scatters from leaves and water and the like, we'll get varying degrees of polarization and varying orientations of that polarization. And we can't sense that, but butterflies use the degree of polarization and the orientation of polarization to help them figure out how all the leaves are oriented so they can fly through bushes and navigate. And octopus and many cephalopods and other animals underwater use the polarization of light scattered from the particles in the water to hunt for little transparent organisms that are trying to hide from them, like worms that they want to eat, diatoms. So there's many uses for this polarization information in the animal kingdom. So let's look at the Meinel Optics building. Okay? Here's an intensity image. And the whole front, the north face of the building is windows, all tilted at different angles. And when light reflects off those windows, it becomes partially polarized. You can also see the football stadium, the upper right. And so here are those Stokes parameter images. Here they're in a grayscale. Here I've used the color scale. These are the same data sets, this and this, just shown two different ways. And so when I look at the building through a horizontal polarizer and through a vertical polarizer, and I subtract them, when it's equal, over here it's gray. And when there's more horizontal polarized light, it's brighter. If there's more vertical polarized light, it's darker. And it's kind of hard to see in here quantitatively how polarized things are. So I also have this scale over here, and you can see that actually in this image, it's all positive. There's more horizontal polarized light, and there's really hardly any place where there's more vertical polarized light. And there's also some light polarized at 45 degrees. And when you combine these, you'll find the actual orientation of any particular pixel. Okay? And so I can now convert that into well, I can also look at the orientation. And so here we have the orientation in grayscale. Here we have the orientation in color. And most of my orientations are in here around horizontal, or close to 180 degrees, because 0 and 180 degrees are the same for the polarization orientation. If you rotate a polarizer by 180 degrees, we get the identical performance. And so we have this polarization information here for the building. And here we have represented in terms of lines. These points are locations where the image is unpolarized. And then the longer the line, the higher the degree of polarization. And so you can see here there's a tendency for this light from these windows to be horizontally oriented. But depending on the orientation, different windows are polarized to different degrees and with different orientations. And so here we show in three colors, 470, 660, and in the near infrared, 865 nanometers, a composite image of those three wavelengths, which are the three wavelengths that we're measuring. And plants are very reflective in the near infrared, which is why these green trees appear red here, because they're actually brighter in the near infrared than they are in the blue and the red that we're showing. And so we can see here the degree of linear polarization, and where this is blue, the blue light is more polarized. Where this is red, the red light is more polarized. So we have different polarizations at the different wavelengths. And this is showing the angle of polarization and its wavelength variation. And so I now want to show you a picture of the back side of the building. We have all the windows on the north, because in Arizona the sun is so bright. And the whole south side of the building has no windows for this wing of the building. And so I want to show you a a time sequence so you can watch the polarization during the day. And so we're going to look from 8 a.m. to 3 p.m. at the intensity. Then we're going to look at the degree of linear polarization, and then look at the angle of polarization. And you're going to see how much this polarization depends on where the sun is in the sky, or what clouds are in the sky, or these other conditions. So it's something that's very dependent on the environment. So first, just to familiarize yourself with the image, this is the intensity during the day. Is it running? So you can see the shadows moving. When we're noon, the shadows are going to be this way. We're looking towards the north. We have some clouds coming and going during the day, and then we get here to the end of the day, the end of the data set, and it's stopped. So that's the image set, the building, the parking lots, trees, the sky, a little hemispherical planetarium dome here. And now let's look at the degree of polarization. And so before I start this animation, we've used what we call here a bright band color scheme, so we can tell very easily if it's red, it's below 10%. If it's purple, it's below 20%. If it's cyan, it's below 30%. Green, orange, and then if it's brown, it's above 50%. So I have 50% polarization up in my sky. I've got some areas down here that are nearly unpolarized. And then we've got lots of areas here that on the building are 30%, 20%, in the teens. So there's quite a bit of polarization in this scene. And right now the sun is off in the east. This is early in the day. And now we'll run the movie. And first, if you look at the sky, you see the sky is now less polarized. We're getting later in the day. The sky is getting more polarized. But there's this circular pattern moving across the sky as the light is unpolarized, close to and opposite the sun, and has a maximum degree of polarization at about 90 degrees from the sun. And then if we look down here, we can see these cars are obviously cars when we look at the degree of polarization. They stand out against the asphalt, and we can sort of see the shapes, very clearly. The contrast is very different than the intensity image, but the information that it's a car is obvious. If we look in the sky for the clouds, here the clouds are less polarized. Here's some clouds. We see that they're red. So the clouds have a much lower degree of polarization. They're not unpolarized, but they tend to have a low polarization. And now last, if we look at the building, here you see we're in the teens in the polarization. We're in the middle of the day. And now getting towards the end of the day, something happens right in here where the building becomes very polarized. So here it's less than 10% polarized, and down here it's about 25, 30% polarized. And what happened? It's in the shadow. Yeah. And when you're in the sunlight, it's about 80 times brighter than when you're in the shadow. Depends on the shadow, but it's an enormous difference. And so this area is being illuminated more by the polarized sky. And this area, which is much, much brighter, is being illuminated by the unpolarized sunlight. And this is the shadow of the library building, which is across the street. And so polarization is actually very useful for looking into shadows and seeing details of things in shadows. And it's one of the applications that has proven very valuable. This is that same data set. This is in grayscale, so it's much harder to quantify, but it's a much more pleasing image. It's not so jarring as the bright bands. In the middle of the day, the polarization is pretty low, and then here we see that shadow. These cars are in the shadow, and a high degree of polarization there. And of course, we have the angle of polarization. And what we're going to see in this angle of polarization, which right now, early in the morning, is in here at this angle around 100, 110 degrees, is that as the sun moves across the sky, the polarization is going to rotate as well. And it's not going to rotate perfectly uniformly among all the different objects, but if you watch it during the sky, you'll see the general tendency of the polarization to rotate all day long. So let's look at this angle of polarization. It's now purple, and now it's maroon, it's coming red, yellow, green. And so this overall polarization state is rotating all day long. So unlike color, something's green in the morning, is green at noon, is green in the evening. The polarization is far more variable, and so it's conveying different information. It's conveying information about refractive index, it's conveying information about orientation, it's conveying information about texture. So when we see color, we're seeing quantum mechanical bond energies. What energy is being absorbed and what energy is being scattered depends on the energy levels in those molecules. But when we see the polarization, we're seeing what direction are those electrons oscillating that are radiating the light. And so if they're oscillating randomly in all directions, we're going to scatter unpolarized light. But if we have a very highly linearly polarized pixel, the electrons in that material are predominantly moving in just one direction and are radiating highly linearly polarized light. So we have a lot of orientation information in these polarization images that we don't have from color. If you see a particular color, it's very hard to infer the orientation of something from its color alone. You need to see the shape, but the color hardly tells you anything about the orientation. So the instrument that we measured these images with is called ground MISPI, and it's what we call a dual photoelastic modulator polarimetric camera, and it uses two crystals that are called photoelastic modulators, and we run sound waves through those crystals and modulate the polarization state at very high speed. And then we have a high-speed detector that's reading out one row of pixels. And if the light is polarized, it gives us a particular Bessel function shaped curve. And if the light is unpolarized, there's no modulation, and we get a constant signal. So based on whether it's constant or a small amplitude or a large amplitude, and these are the particular 20 measurements that are taken, we can measure the degree of polarization. And we have two polarizers back there, one at 0 degrees and one at 45 degrees, that make this polarization measurement. So there's one row of pixels that has a horizontal polarizer, and then we need a space of a couple pixels, and we have another polarizer that's at 45 degrees. The photoelastic modulator is a crystal of fused silica that's driven with a sound wave from a photoelastic transducer. So we're shaking this piezoelectric transducer, and we're setting up sound waves in this crystal. And those sound waves are at its lowest mechanical resonant frequency. So if the crystal is in its neutral state, if it's off, it has a particular octagonal shape. But the sound wave causes the crystal to first compress in one direction and then compress in the other direction. And when we compress the crystal, it becomes a retarder, has a different refractive index in these two directions, and so it modulates the polarization state. And with these crystals, we can set up a very loud acoustic wave. They have a Q of about 10,000, which means the wave builds up for about 10,000 cycles, gets to a pretty large amplitude, and we can actually get these crystals not just a quarter wave or half wave retarder, but we can get them to about 3 waves of retardance by turning up the sound wave to very loud amplitude. And so the light comes in from the telescope. It goes through a quarter wave retarder. There are 2 of these crystals there's another quarter wave retarder, and then we have the polarizer. And the effect of this combination of quarter wave retarders and our polarization modulators is that if there's linearly polarized light, it causes the plane of that linear polarization to oscillate. So if it's unpolarized, well, it doesn't oscillate. But when it's linearly polarized, it rocks back and forth. And then when it gets to the polarizer, that produces this modulation. And then there are our spectral filters and the detector that reads this out. And this just shows a ray trace, the optical design. There is a 4-mirror telescope here and our quarter wave plates and these large crystals. They're almost as large as the rest of the optical system. And there's a side view and a top view. And we have a large field of view here. So from space, it would be able to pick up a large swath of the Earth. And so this was the first one. It proved the concept of these photoelastic modulators, and it gave us this portable instrument we could bring around and collect lots of these polarization signatures. And this program ran for 3 years. It was very successful. So we got another 3 years of funding. When I say us, I mean the Jet Propulsion Laboratory, who's responsible for the instrument, and I'm a co-investigator. And we built a version that we could put on a high-altitude airplane. It's called the ER2, which is and it flies at about 62,000 feet. And so this is flown multiple times to collect space-like data from high altitudes. And it measures intensity at all of these different wavelengths, and it measures polarization at 470, 660, and 865 nanometers. And all of these wavelengths and these polarization measurements were chosen to optimize the sensitivity to measure aerosols from space. So we have clouds, we have dry air, and in between we have air, that has small amounts of water droplets or small amounts of soot or small amounts of other particles in it, and we're very interested to measure those particles, measure their distribution. And by measuring polarization, we can measure these aerosol properties about 10 times better than we can measure just by making intensity measurements. So the satellites in orbit now that measure the aerosols from a set of wavelengths, but by measuring polarization, we can measure these aerosols now in order of magnitude more accurately. And then we got a third 3-year contract, and we're able to extend the instrument out to 2 microns. So we added several additional wavelengths here that give it a better range and accuracy for these aerosol measurements. And this started flying in October of 2015, this Air MISPI 2. And it has flown over California, Texas, and recently it was sent to Namibia for 30 days and took data in Africa. And so this is a picture of Air MISPI mounted on the ER2 aircraft. And it has flown more than 20 times to collect these polarization images and get the data sets so that the scientists can work on the data reduction, demonstrate for NASA that there's a genuinely useful data product that will answer these important questions. So finally, MISPI was optimized for making these space-based remote sensing measurements of aerosols. The aerosols are one of the major sources of uncertainty in the magnitude of the climate forcing from the sun. So how much of the sun's radiation is absorbed from the Earth, how much of it is scattered back into space, tells us the energy balance. And if the Earth is warming up, then less radiation is going into space and more is staying on the Earth. And we have a very good idea how much light scatters back into space from the clouds. We have a very good idea how much scatters back from the ocean and from the Earth's surface. But the aerosols in the atmosphere are much more difficult to measure because they're hard to see. They do scatter light, So we have the background of the Earth or the background of the ocean, and to separate out what part of that is the aerosol and what part of that is the Earth, well, this is where the polarization comes into play, because the polarization of the aerosols can be calculated pretty accurately from theory. And so this gives us an ability to separate the aerosol from the Earth background signal and measure it more accurately. And so these airborne particulate matter is also known to be a major health hazard. So when there are particles in the air, like soot from power plants and from industrial processes and oil refineries and the like, we know that this is bad for human health, but we don't have good measures of how bad it is. Okay? So here are some examples of the aerosol measurements to give you an idea. This is a conventional RGB image, and you can see some agricultural fields here in the Central Valley in California. And as I mentioned, in the near-infrared, so this is blue, red, and near-infrared, vegetation is extremely reflective. Okay. And then here we have a degree of polarization image looking down from the plane, and we can see areas where there is more blue scattering and areas where there is more of this 660 and 865 nanometer scattering. So you see the wavelength dependence, and where this is black, this is unpolarized, nearly unpolarized, and that's mostly corresponding to these clouds. So we have the clouds, we have the fields here, and then with the polarization, we can begin to pull out, well, notice how blue this is, right? Well, the sky is blue. And so most of this polarization here that we're seeing, this blue, is coming from the sky. And so we're able to separate the skylight from the background Earth light and gain information on the aerosol content. And then we can choose a target area, and as we fly over, we can rotate the instrument, take a series of images of the same area, and when we reduce the data, we'll be able to get the aerosol content at different depths. So we can build up, the aerosol near the Earth, the aerosol up in the stratosphere, and get the three-dimensional distribution. And here's an example where we have scanned the camera continuously. So here we're looking at 60 degrees, and then 30 degrees, and then down, and then scanning backwards. And we're moving forward slowly as we do this. And so from this data set here, we're looking at a cloudy area, okay, over the ocean here. But when we start looking away from the sun, so here we're looking towards the sun, the reflected light from the sun, and here we're looking like back with the sun in one direction, and the instrument is measuring back, you can see that we go through the angles where rainbows form and beyond those angles, and that there's an enormous amount of polarization information from this cloud light at certain angles. So in these angles, the polarization information doesn't depend on wavelength very much. And then when we go through 140 degrees from the sun where the rainbow formed, all of a sudden we get a large amount of information. So these are the angles that are going to be most useful for us. And the orbit is chosen so that it looks at the Earth near these angles to get the largest resolution between the different wavelengths on the polarization signatures. If you've looked down from airplanes, you've probably seen the glory, which is a little bright spot that follows the airplane along. Okay, and so here we have the polarization image, and in the intensity image here, you can see the glory here faintly, and there's a sort of spectral rings that form around the glory. When we look at the glory in degree of polarization, there's lots of information. And so this is the type of information that's telling us about the droplet size, the droplet refractive index. And so this is an example of how much more information there is in this polarization image than there is in this intensity image about the aerosols. Okay. This is the information that we're using, the polarization variation with angle and with wavelength. And so this has been approved now, and $30 million has been awarded to Jet Propulsion Laboratory to build a new version of this instrument. And so it's now called the Multi-Angle Imager for Aerosols, or MAIA. And it will fly about 2019. And the focus of the instrument is to measure these airborne particulate matter, which is a well-known cause of cardiovascular diseases, heart attacks, low birth weight, lung cancer, and premature death. But the toxicity of these different particulate matter classifications, like there's a 2.5 micron classification and a 10 micron classification, is really poorly understood. So MAIA is going to fill this gap because it will be particularly focused on areas of metropolitan areas like Los Angeles and New York, and some places with lots of pollution like Delhi and Sao Paulo. And they'll be able to correlate the aerosol histories over those places with the health records that are collected from hospitals, with the public health information. And so this will help close the loop to understand the relationship of pollution and particulate matter and aerosols and how that is correlated with disease incidence in these metropolitan areas. Here is a picture of the instrument in space. It will have two of these polarization cameras that will go side by side to double the swath. And this are going to be sampling these metropolitan areas with sub-kilometer spatial resolution. And it's very similar to the Air MISPI. It's just the next generation. And we're not allowed to make many changes once we demonstrate these instrument concepts. So in conclusion, there's a large amount of information in polarization scenes. And my laboratory has participated with the Jet Propulsion Laboratory in the development of a series of these imaging polarimeters for remote sensing. And now finally, this MAIA instrument is now in design and construction and will be applied to the monitoring of aerosols in metropolitan areas for health research. And so one of the things that's going to be needed 5 years from now and beyond are scientists that understand these polarization images. So there's many scientists now in remote sensing and its application who are trained in multispectral and hyperspectral imaging, because those satellites have been in space for 30 years collecting data. But there are not scientists trained to interpret the polarization images. And this instrument is specifically been designed for these health applications, but it will also have many other applications in remote sensing, studying the volcanoes and weather systems and the ocean, the health of crops and the like. So this is just one application of the data and the focused application that got funded. So I'll show you one last set of polarization movies here. This is from the roof of this building. Out front, there's a bus stop in front of the building, and there's some very interesting polarization on these metal surfaces. And this was during a carnival called Spring Flings. There's also a Ferris wheel out there. And so, are there any questions? Thank you for your attention.
Questions, yeah.
So it's not too difficult to conceptualize the polarization of waves of light, but what would it mean to talk about the polarization of a single photon?
I like your question. What would it mean to talk about the polarization of a single photon? Since it takes me 4 measurements to measure my Stokes parameters, I can't measure the polarization of a single photon. So, I mean, that every photon has, some uncertainty in its position and its momentum and its polarization state. And so there is uncertainty, in the polarization of any single photon in terms of my being able to determine its polarization state. Now, maybe I can produce 1,000 photons and they all have the same polarization, and now I can infer that the succeeding photons from that same source will have the same polarization. But regarding that single photon, if I put in a polarizer, well, it makes it through or it doesn't. And if it doesn't make it through, I don't know that it was there, right? And if it makes it through a horizontal polarizer, well, it might still be polarized at 45 degrees or almost at 90 degrees and have some probability to be transmitted. So, I mean, you asked a really good question about, the information and what's measurable. And that's my answer.
So would the same design work for circular polarization or any other? Would you only have to change the filters, or would the entire oscillator have to be constructed?
You can modify the design for circular polarization. In these natural scenes, the circular polarization tends to be below 2% and doesn't convey a lot of information. And transmitting information down from space takes energy, power, and cost, and we can't transmit all the information that we could measure. So we actually have to restrict the number of wavelengths and restrict the number of polarizations down to what we can afford to transmit down. So the circular polarization is very interesting, but it's not a significant component in these remote sensing instruments, or we'd also be measuring it.
There's no problem taking like a whole spectrum. It's just that restriction of how much you can.
Right. And this instrument has bandpass filters, but you can make polarization measurements with spectrometers or Fourier transform spectrometers and measure the polarization as a function of wavelength. And this is done, one of the best examples is in measuring solar magnetic fields. There's a polarization in very narrow, 100th of a nanometer bands in the sun. And by scanning wavelength and measuring polarization, you can map the magnetic fields on the sun. So that's an example where they make that kind of spectral polarization measurement. They do it in chemistry a lot also. Yes?
Is it possible to use your instrument in a ground-based configuration to look up at the sky and see scattered polarization? So what's the advantage of flying it?
Well, the advantage of flying it is we want to, we would love to map the entire Earth for aerosol, on a regular basis, like twice a week, like multispectral measurements are made now. And so looking up, I can get information on the aerosol content, and we have had several cases where we flew on the ER-2 aircraft, and we had ground MISPI on the ground, and we synchronized their measurements. But if we want to map the whole Earth, if we want to map metropolitan areas, then space is the way to go. Yes?
Like might be made for much smaller but higher resolution, like more localized images to measure things like polarization of like mature cloud droplets or like aerosols that cause like
Yeah, absolutely. And so there's quite a few similar instruments that have flown in airplanes and studied cloud systems and studied scattered light from water. You can see oil slicks on the water very nicely with the polarization, and there's airborne instruments that are specialized for that. Yes?
Does it work similarly to LiDAR, or is it different?
Well, LiDAR is active. And from space, to make these large-scale measurements, we need a passive system with solar illumination. But there are plenty of systems that illuminate and measure the polarization. So you can use a spotlight with polarizers, illuminate a scene with circular polarized light, and get great information in something like that from the circular and linear polarization. So we have active polarization measurement systems also. But
engineering and electrical and computer engineering, and optical sciences incorporates people from chemistry and biochemistry, from medical imaging, radiology.
You asked me to do the microphone.
Okay.
You can just keep going, Jennifer. I got it off the other mic. So.
Okay, great. All right, well, I was just welcoming you all and talking about how interdisciplinary optical sciences is. And so I'm going to give you very briefly a discussion of optics in biology and medicine. I'll briefly talk about tissue optics because the way light propagates through tissue is a little different than other materials that we commonly think about in optical sciences. And then I'll go through 3 types of imaging. And of course, resolution is always relative. When we talk about ultra high resolution, we mean generally a micron or less. High resolution imaging, which is maybe 10 micron resolution, and low resolution imaging, which is maybe about 1 millimeter. So you can actually image on a variety of spatial resolution scales with optics. And then finally, I'll end up with therapy because you can use light not only to image, but you can use light to actually affect some change you want. But I'd like to start off with the fact that, in optics, we are here for an application. So I spent a lot of time going around and talking to biologists and doctors about what do you want? If I could give you an ideal imaging modality, what would it be? And they tend to tell me the same things all the time, regardless of the very specific application. They want cellular level imaging or imaging at very small tissue microstructures, both structural, anatomical, and functional imaging. We want it to be inexpensive, don't let it harm the tissue, minimally invasive, we don't want to have to poke needles in you unless we need to, deep penetration, high contrast, easy to use, intuitive display. Well, okay. We can't give you all of those all the time, but we can give you most of those most of the time. So new optical imaging modalities really do promise to do this. The advantages of optics is, as I mentioned, you have a variety of spatial scales. You do have sensitivity to tissue structure and some inherent ability to measure function as well. It's relatively inexpensive compared to something like magnetic resonance imaging. It's very inexpensive. And there is no ionizing radiation, unlike you have with, say, a CAT scan. But there's some challenges. Light typically does not penetrate more than a couple millimeters into tissue. There are some, alternatives to that, but usually the penetration depth is poor. You typically have a small field of view, so we oftentimes end up with this soda straw dilemma, where we can come up with a high-resolution imaging technique, but people talk about it like trying to find the cup on a golf course by looking through a soda straw, right? You could find it eventually, but it might take you a long, long time. So we do have this problem with small field of view offenness. Often the endogenous contrast is poor, so we have to come up with tricks to get around that. And it's not always intuitive to look at, especially for physicians who tend to be trained in a very specific way. We're throwing new information at them, and so we need to give them some training. All right. So that is background. What is tissue optics? It's really a consideration of the unique ways that light interacts with living matter. Now, I just finished telling you that light typically doesn't penetrate very deeply. Well, actually, it does. This is one of my favorite pictures of one of my mentors, Dr. Bruce Tromberg, who took this picture. He just stuck a flashlight in his mouth and took a picture and said, look at all the light coming, especially through my sinuses. So light can propagate long distances, but it's very fuzzy, right? You certainly don't have cellular level detail here. So it's interesting, light operates in many different modes, from high resolution to low resolution, depending on the distance it goes through. Okay, tissue optics is also a consideration in the unique needs and constraints of working with living matter. So I'll give a couple of images here. On the left, this is a surgical image in the peritoneal cavity, and this image on the far left is what it would look like to the physician just looking through an endoscope. And all you see is a bunch of pink. Okay, but if you put a special dye on the tissue that binds to the folate receptor, which happens to be this structure on the cell surface that's upregulated in ovarian cancer, all of a sudden these little cancer spots pop out at you. Okay, that information was always there, but we had to have a special targeted dye in order to get it. Hopefully none of you have experienced this. I know I did back in the bad old days when we didn't know how to stay out of the sun. Light can harm tissue. So if your point is to image, you need to be really careful to avoid this and avoid photobleaching and other things which aren't quite as dramatic. If you want to kill tissue or cook tissue, you can. All right. That's not usually the best way to do it. Okay, so tissue optics, the most important thing in tissue optics is scattering, and it's really one thing that differentiates working with tissue from people who just do a lot of lens design and tend to work with clear materials or non-scattering materials. We work with highly scattering. You are highly scattering, and that's typically what blocks the light more than anything else. And not only that, but tissue is composed of structures from nanometer to meter scales. And so the scattering is very complex. I've shown a tendon here. Well, a tendon is composed of bundles that are composed of smaller bundles that are composed of, fibrils that are composed all the way down to collagen molecules. And they're all twisted together. So you can't just use something like, Rayleigh scattering to describe tissue scattering. It's more complex than that. So we tend to have very difficult times really coming up with an exact model of light propagation through tissue because it is so complex. It is theoretically possible to do that, and people have tried, but usually it's not practical. We tend to just think of the bulk properties of tissue. And so we'll tend to think of a bulk scattering coefficient in tissue that talks about how light spreads out. So the scattering coefficient will talk about how it scatters, but you also need to know something else, which is what is the average cosine of the scattering direction. So it turns out that tissue scatters very strongly, but it tends to scatter in the forward direction. And that has strong implications for both therapy and imaging. One thing to remember, if you remember nothing else, scattering monotonically decreases with wavelength. So when we're trying to image deep, we always like to use longer wavelengths. And if you look at this, skin is red. So this is kind of the bulk scattering coefficient of tissue. This is actually the reduced scattering coefficient that takes into account the forward scattering properties. And interestingly enough, if you look at a Rayleigh scattering model and a me scattering model and look at their combination, sure enough, yeah, actually, as we would expect, tissue is so complicated that in the bulk, you can just kind of think of it as being a combination of Rayleigh scattering at the shorter wavelengths and me scattering at the longer wavelengths, and look at it that way. So that simplifies things greatly if we just look at the bulk properties. What does this do? Well, if we shine a laser beam into some sort of, tissue phantom and look at how the light spreads out, you tend to have 3 different areas for approximately 100 microns deep into tissue. And of course, this varies on the tissue, but in general terms, you have something we call ballistic regime, where basically all the photons are still going straight ahead. And this is where you can do microscopy, really ultra-high resolution imaging, where you get very, the photons are well behaved. You can, pretty much understand the way that they propagate, and so that's fine. Then up until about 1 millimeter, you get this area of increasing photon scattering where those ballistic photons become rarer and rarer and rarer. And there are imaging techniques that go to heroic lengths to separate out the ballistic photons from the ones that have been multiply scattered, and that's where I tend to work, things like optical coherence tomography. Beyond 1 millimeter, we enter this regime called the random walk, where, all hope is lost. There are no more ballistic photons. But in that regime, the light behaves just kind of as like a uniform diffusing wave. And there you can use a different set of optical imaging techniques that don't try in any way to understand how the photons propagate. We just know probabilistically how they do, and you can actually use that then. So these 3 different regimes are what we typically do, and that's typically what drives the development of imaging techniques. Now, you said scattering. What about absorption? Of course, absorption is really important too, but interestingly enough, for imaging, scattering is usually more important. Absorption. What are our major chromophores, things that absorb light in tissue? Well, you have melanin, and of course, for all of us, that amount of melanin determines our skin color, and that varies quite a bit from person to person. We all have blood, and so we have hemoglobin and oxyhemoglobin. slightly different absorption spectrums there. That's how things like pulse oximeters work, is looking at the difference between oxygenated and deoxygenated hemoglobin. And then one that most people forget is water, which actually is a very strong chromophore once you start getting into the infrared. If you look at the range between about 700 and 1100 nanometers, absorption is low. And that's typically what we call the optical window, where you can image pretty deeply into tissue. So things that do deep imaging tend to work in that range. Fortunately, it's also longer wavelength, and your scattering is pretty low. So a lot of optical techniques will work there. Now, you have the competing problem, which is that, as you, I'm sure, have all had, when you talk about optics, your resolution is always better the shorter wavelength you have. So you tend to give a trade-off as you go to longer wavelengths. You give up a little bit of resolution, but you get a lot of depth of imaging. All right, so those are the two major things that affect just about every imaging technique. We also have fluorescence. You're fluorescent. I'm sure you've noticed that if you've been under a black light. Different properties of tissue are fluorescent, and your fluorescence changes in disease. And so fluorescence in tissue, though, is more complicated than you might think, because you've got a lot of types of molecules in your body that fluoresce. And so if we shine light on the tissue, we've got all these different types of fluorophores at different depths in the tissue. They may emit light, fluorescent light isotropically. A certain amount of that light is captured back by our system and measured by a spectrometer. What usually we want to get out of fluorescence imaging is a measure of fluorophore concentration as a function of position, and that's extraordinarily difficult to get back. However, if you just want to kind of get a general depth-weighted measure of how fluorescent tissue is, then that's easy to do. This is one thing we do a lot in fluorescence, which is we take a look at excitation wavelengths, so going maybe here from the ultraviolet up into the green, and then we look at the emission that comes back. You're really, really fluorescent in the ultraviolet. So this would be one case where if you, and by the time you get to the green, you're hardly fluorescent at all. There are some fluorophores, even in the red, you're slightly fluorescent, but not very much. So this goes against our ability to work in that optical window, because you tend to have fluorophores in your body that tend to be most fluorescent only when working with ultraviolet and blue wavelengths. Okay. This calls for maybe wanting to use something like exogenous dyes, where we can put dyes into the body that do fluoresce in the near-infrared wavelength. And there's actually one that's FDA approved called indocyanine green, which is called that because it looks green, right, which means it absorbs longer wavelengths. And so it's actually fluoresces in the near-infrared. Unfortunately, it's non-selective. If you've seen these beautiful images of cells, fluorescence microscopy, all this stuff is targeted. These are specific chemicals that bind to specific types of receptors on specific cellular constituents. None of those are FDA approved. So we kind of have this problem where our FDA-approved stuff tends to be very bland and nonspecific. It can be useful, but there are some people who are trying very hard to get some targeted fluorophores approved. Okay, so I mentioned those three different imaging regimes. And so here we go. If we put all optical imaging techniques on a plot of resolution versus depth of penetration, they tend to go in a diagonal line, right? The better resolution you want, the lower depth of penetration you have. Down here we have our ultra-high ballistic regime techniques. Here we have our kind of in that intermediate area where we're trying very hard to get out those ballistic photons. And then up here we have a technique that can work with that diffusing wave. So let me just go through those pretty quickly, starting with microscopy. I put this up here because I think most people, unless you've had an opportunity to work in modern microscopy, microscopes have gotten really very sophisticated nowadays. And this is a typical layout of a microscope you might find in a modern biosciences lab. Your sample goes right here, and you can either illuminate it from the bottom, call that transillumination, or you can illuminate your sample from the top, and we'll call that epi-illumination. And in any case, the light that's either scattered, transmitted, or fluorescent light comes up and can either go to a camera or to an eyepiece. So what do you see? The most common type of microscopy still is what we call bright field. That's transillumination. You're illuminating a thin sample from the bottom, and you're looking at what transmits through. The only mechanism of contrast here is absorption. And so this is your very typical, hematoxylin and eosin stained tissue slice that you see. This is the gold standard. So everything we do in optical imaging, the physician is going to compare to this. Okay. Now, of course, the great disadvantage of this is that you have to take the tissue out. You have to embed it in paraffin overnight, at least. You have to slice it very thin, and then you have to stain it. There's no way you can get this done in less than a couple of days. And then you have some pathologists look through a microscope, and this is the gold standard. You can see the cells. You can see the tissue architecture. You can see larger structures. This happens to be a section of a normal mouse ovary and an adenocarcinoma, and people spend their careers interpreting how these structures change. But there's a lot more you can do with microscopy. If you just take that same system and instead of putting the pickled slice underneath there, you put a whole chunk of tissue and you illuminate it in what we call bright field or epi-illumination, then you can actually start seeing some things as well. This is a small tumor growing in normal tissue. Now you can probably see, even though the lights are kind of bright, but you can probably still see there's very abnormal blood vessels in here. So the mechanism of contrast we're getting here is both absorption from the blood mostly, but scattering too, because of course the light has to scatter around and come back up in order for you to see this. So this is pretty common, and actually this is just how endoscopes work, right? They just stick, an endoscope into the body, and they're just looking at the light that reflects back, just white light endoscopy. And so this is also something a physician will understand. We don't have quite the high level of resolution because the scattering has blurred it all out, but we get lots of information. In honor of Dr. Chipman, I'll put up the fact that polarization microscopy is also really commonly used, and that also can be done in transillumination. Some tissues, like bone, are very highly birefringent, and they will alter the polarization state of light, and you can look at that and look at bone growth. You can also use polarization in a larger reflectance mode. This is an image of skin, and if you looked at this, you'd probably have no idea that there was a basal cell carcinoma that was lurking there underneath the skin. But if you look at it under cross-polarized light, you can clearly, at least people who are used to looking at these images, there is a cancer right here that's actually clearly demarcated. So again, polarization is also one of those aspects. There's lots of other things I'm not going to cover that some of you all may be familiar with, where you can do things with dark field and phase sensitive and other things like that. Fluorescence is another way. So we can take a look at fluorescence like I showed you before. And this is epi-illumination with exogenous dyes and cells. But we can also have fluorescence that we do in vivo or with chunks of tissue. These are pieces of tissue. This is a white light image. And this is images of that same tissue under different excitation wavelengths, all the way from the ultraviolet into the blue. And you can see how they fluoresce differently. I think it's really amazing to me that this chunk of fallopian tube over here that fluoresces so strongly in 280 nanometer light is almost completely absent in 320. So the fluorescence properties of tissue change from tissue to tissue and from healthy to diseased. So that's your kind of standard microscopy. And if we want to get to more advanced, you've probably heard of some of these before. For instance, confocal microscope. All these techniques I've shown you before take all the light that comes back, and it just integrates it. Doesn't matter from where in depth the light came. Your intensity is basically the same as a function of z. What if we want to be able to try to reject light that's out of focus? That's exactly what a confocal microscope does. A standard epi-illumination confocal microscope will work by taking a point source, oftentimes a laser or light coming out of a fiber, focus it onto the tissue. The fluorescent light that comes back is diverted by a dichroic beam splitter, and only the light really that originates from your focal point within your numerical aperture will make it through the pinhole to the detector. It does a good job of rejecting light that's out of focus. And so with that, all of a sudden, you can start getting microscope images that aren't just, some two-dimensional integration. You can start getting these beautiful three-dimensional renderings, such as shown in this group of, tumor spheroids here. This is a rendering of many, many different depth sections put back together. And we can put that into the body as well. There's a lot of work going on here. Dr. Artemitro is working on this confocal microendoscope. Here it is. It's a couple of millimeters in diameter. It can go through the instrument channel of a standard bronchoscope or colonoscope. And it's actually quite sophisticated. That's one of the great things about being here, is that we can come up with these nice lens systems that have focusing and everything in them. So that's confocal, and we'll take it even further. I don't have time to go into the mechanisms, but multiphoton microscopy, the essential idea is that you get light that's so squeezed in time and space that you end up with nonlinear phenomena. You can have two photons that are absorbed and by a molecule that essentially thinks of it as one photon at half the energy. And so you end up then with fluorescence that's at a shorter wavelength than your excitation. You can also have two photons that are combined in a scattering event, and we call that second harmonic generation. Turns out that there are crystalline-like parts of your body, collagen being most notable, that will generate second harmonic. This is great because collagen is one of those things that changes most quickly during disease. And so we can generate really very beautiful images of tissue where we can start getting both second harmonic generation, it's shown here in the red, and third harmonic generation shown here in the blue. And the great thing is that this stuff we can do in vivo. So we sometimes do rather heroic things where we try to take existing big tabletop instruments. We've got a multiphoton microscope that's a 4x8 optical table, and we can put anesthesia and warming, and we can do sterile survival surgeries on small animals and image them that way. When we do that, we can get really neat time serial images. This is looking at the two-photon excited fluorescence and the second harmonic generation over time. We can see, if we zoom in on that, how as we go from normal healthy tissue, we have low fluorescence, nice linear fibers, to when we have a tumor here, we've got lots and lots of fluorescence in these tangled fibers. We want to put that into people. This is going to be a little bit of a trick, but that's the type of thing that we do here as optical sciences, is figure out how to take that tabletop instrument and make it something we can bring into the operating room. All right. So that's your ultra-high resolution techniques. All of those had resolutions of a micron or less. In that middle range, there's something called optical coherence tomography. Who here has heard of OCT? Okay, a lot of you. That's great. It's getting more well known. It's actually now commercialized for looking at the retina, but not much else yet. In OCT, we are trying very hard to extract out those ballistic photons. And we do that by basically timing how long it takes the wave to go into tissue. So it's the optical analog of ultrasound. We take an electromagnetic wave, it goes into the tissue, it comes back out, and we time it, and we know by the timing how deep it went. And we know that if it takes too long to come back out, that obviously it was multiply scattered in the tissue and we reject it. That's the way that we are able to image up to 1 millimeter deep with nearly the same resolution that you can get with those ultra-high resolution techniques. So it's really outstanding for mapping microstructure of tissues. Typically, really, from a practical standpoint, it works more at the 10 micron resolution level, and it can image things like, precancers in tissue, stent struts in artery, and this is a mouse colon that's only 400 microns thick. Here's another example of that where really we can get some nice exquisite resolution. This is 15 millimeters long and 1 millimeter deep. We can get really exquisite resolution about what's going on at the boundary of the mucosa in the colon, which is where disease occurs. Okay. We can also put it together because with OCT you can get three-dimensional images. And so here's just a comparison of a normal ovary, a benign adenoma, and an adenocarcinoma, which is the bad cancer that we're worrying about. What we're hoping is to start making images like this that look a little bit like those pink and purple H&E images you saw. And of course, they're not pink and purple. They're grayscale. But we're starting to see things not quite cellular level, but at that anatomical scale that we're hoping physicians and pathologists will accept. I mean, clearly, you can tell this is diseased because even you can see this is different than that. Right? And then furthermore, what we can do that you can't get with H&E histology is that totally non-destructively and minimally invasively, we can get complete depth sections into the tissue. So we can look deeper and deeper and deeper, and we can see that whole three-dimensional structure, right there in real time with, without doing any damage to the tissue. So not quite as high resolution, but a lot of benefits. And then we want to be able to take this and put this into small endoscopes. So again, getting to one of our things we do here as optical sciences majors is take that big stuff that's out on a table and put it into something that can go into the operating room. This is we're building a 800 micron diameter endoscope that has both a fluorescence imaging channel, an illumination channel, and an OCT channel. Here it is next to just a model of a female reproductive tract. Here it is. We're building it. There's the lenses, fiber bundle. We're building it under a microscope. And we do take these things into the operating room. And so that's our physician collaborator here. And this is one of our slightly larger laparoscopes that we have going into the belly of a woman and looking at her ovary here. So. Okay, I want to cover a couple of other imaging modalities, and I'm mindful of the time here. There's a couple other ones that you should know about. Photoacoustic imaging. This is interesting because photoacoustic imaging combines optical and ultrasound together. So its mechanism of contrast is just absorption. So it works really well for mapping blood vessels or maybe things like melanoma where you have a strong absorber. If you put a pulsed laser beam into tissue and you just flood the whole tissue with light, if there's an absorber there and the pulse duration is short enough, you'll actually cause a shock wave to be generated. That absorber will absorb the light, it will heat up, it will expand, it will cause a pressure wave to go out throughout the tissue. And you can measure that back on the surface of the tissue with an array of detectors. And with that, you can really get some nice, exquisite images that show you nothing to do with tissue scattering, but actually tell you all about absorption. So this is actually a small melanoma that's embedded in a network of blood vessels. So this is a really interesting technique, and it's a little different than things we've talked about because of its mechanism of contrast. I want to spend a little bit longer on diffuse optical tomography because now we've gotten into that regime where we're just looking at diffuse light. So it's the spatially inhomogeneous optical properties are inferred from surface measurements. So this is inherently an ill-posed problem where we're trying to figure out what's going on inside a volume of tissue by looking at measurements on a surface. So the advantages of diffuse optical tomography are that you can get functional information. If you do this as a function of wavelength, you can get information about the oxygenation state of the tissue or how much water or how much fat is in there. And it's touted as being cost-effective and portable, and I guess it is compared to CT and MRI. I've never seen one that looked all that portable. They tend to still be very large, most of them. But the disadvantage, as I say, is it's an ill-posed problem, and the reconstruction is difficult. So therefore, it tends to be low resolution. Here's an example of a breast imager. So this is designed to, originally the thought was that this would be a replacement for mammography. I don't suppose any of the women in here would rather use this instrument than the mammography that we have to go through right now. But the idea is good, that it would be, simply light-based, no ionizing radiation, and it could be something that could be safer and potentially more capable. So how does it work? We know that if we have a volume of tissue, a sphere of tissue, and we put a light source into it, there's typically this like banana probabilistic path that photons take between the emitter and the detector. And so if we put some detectors around, we know that we've got these probabilistic bananas. And if we move this light source around and keep taking measurements of there, we ought to be able to reconstruct that. Okay, talking about ill-posed problems. The forward problem is easy, right? If you have a dinosaur, you can guess what kind of footprint it's going to make, right? So, however, usually what we're ended up with is the footprint. And we have to figure out what dinosaur it came from. Did it come from the little green one that I showed earlier? A different one? Come from Barney? Or is it the bad? What we're looking for is, do we have something malignant going on here? And that is much more difficult, but people are working on that and they're getting better. And it turns out that really what the diffuse optical tomography has been really quite spectacular at is giving you that functional information. So this is a standard mammography. There's something on there. They don't know what it is. Is that something normal or is it something abnormal? Well, right now that woman would have to go in and have a biopsy taken. If you use diffuse optical tomography, you can get a measurement. It's a blobby, ugly image, right? But you see the hemoglobin is highly, there's a lot of hemoglobin there. It's highly deoxygenated. It's very watery. The scattering size is low, but there's a lot of them. And that's a bad sign. So probably that is a cancer. Actually, unfortunately, this will tell you quite distinctly that is a cancer. And probably the woman should just go straight to surgery. So I think the original idea of this is an interesting case where diffuse optical tomography is morphed in its original intent and has found new ways to be useful. All right, I also want to mention a couple of other diffuse techniques. You may have heard of bioluminescence imaging or dark box imagers. Typically, these are used for small animals where we put an animal inside of a dark box, and you either, inject, it either has a firefly gene, and so you can measure luciferase reaction, or you illuminate it with light and you look at fluorescence. And you can do tomography with this as well. If you get multiple images from multiple different sites on the animal, you can reconstruct it and get some blobby images that will tell you a lot about what's happening with cancer growth or some other targeted item there. I think you're going to get a copy of these slides, so I just put this in for your reference. All these different types of modalities we've talked about, their resolution gets worse, their penetration gets better. What's the mechanism of contrast, and where are they in terms of clinical translation, which is my interest. Okay, we're running a little bit late, but let me just briefly mention, so far I've talked all about imaging, and there's a lot of people working on imaging. It's a really interesting medical problem. It's really important. But you can also use light to do treatment as well. And actually, there's a huge market for that. So what if we want to have light alter the tissue? That's generally a matter of increasing the irradiance or the watts per square centimeter. So you can do that by shortening the pulse length or simply by putting more energy on the tissue. And so I'm going to give you a quick application of port wine stain. Somebody in here may have one, or you may know somebody who has a port wine stain. Mikhail Gorbachev had the most famous port wine stain probably ever. But these things can range from simply very interesting, like what he has, to ones where this lady she has a whole website about loving your mark, to this lady wishes she could get rid of hers. And if she doesn't do something about it, it may impact her eyesight later on because it involves the eye. So how can we use lasers to treat this? I don't know if you remember a long time ago, this laser pioneer, Arthur Schawlow, he used to love to do this trick where he'd come up with his laser gun, and he has a little black Mickey Mouse inside of a clear balloon, and he'd shoot it, and Mickey Mouse would explode, and the outer balloon would be fine, right? So we're going to do the same thing, where Mickey Mouse is the blood vessels, and we're going to kill them. The clear balloon is the rest of the skin, and we're going to leave that alone. And if you want to go on the website, it's always fun to watch people doing these tricks nowadays, too. Yeah. So a simplified model, we have your skin here. This is your dermis. This is your epidermis. Your epidermis has melanin in it, and you want to be careful. You don't want to burn that or you'll scar. Here's a blood vessel. We want to get rid of it. If we illuminate that blood vessel and the light diffuses in, hopefully what it will do is it will heat up that blood, create heat. That heat will diffuse out just a little bit, and it will destroy the blood vessel wall, and then it will be done. So we're targeting oxyhemoglobin because that's mostly what's in the blood vessel. And so which wavelength should we use? You might think blue. The only problem is that it turns out melanin also absorbs very strongly in the blue. And so you actually don't want to use blue. It turns out you want to use yellow light, but because that's still well absorbed, it hits one of these peaks here, but the melanin absorption is lower. So in laser tissue interaction, we're going to propagate light through tissue. The light's going to be absorbed, ideally only in what we want it to be absorbed in, the blood, or if you want to get rid of, say, another type of pigmented birthmark, maybe it is melanin. That target heats up, the heat diffuses through the tissue, and the tissue is damaged. And the one interesting part where things always end up wrapping in biology is the last step I love, and then a miracle occurs, which is where once we heat up and destroy that blood vessel, the body frequently will reabsorb the blood vessel and repermeate the tissue with normal blood vessels. And this is an area where we can't get away from working with biologists because it's important that we understand this step as well. Because there are amazing successes. I mean, this person had an amazing success where they had a very dark port wine stain that was involving their eye and nose. It was starting to become a medical problem. They underwent laser treatment, and it's almost completely gone. This lady had is another example where considerable lightening, although this was just for, probably just for cosmetic purposes. But that's only about 25% of patients. 50% have some improvement, and 25% don't respond at all. And so that's what we'll continue to work on as optical scientists, working on coming up with better lasers, better understanding of how the light propagates through the tissue, and then working with biologists to better understand those biological mechanisms that happen after we treat the tissue with light. So thank you very much, and I'll be happy to take any questions.
You talked about non-invasive imaging. I was wondering how negative photobleaching is for natural fluorophores.
Yeah, so you can bleach naturally occurring fluorophores as well. And so usually when you are doing tissue imaging of endogenous fluorophores, your light intensities are low enough that it's not all that appreciable to see. Photobleaching, although you can photobleach some, it's typically not much of a problem as long as we do things like if we're imaging the tissue fluorescence, we'll make sure we turn off the laser when we're not actually acquiring photons at the CCD and things like that. The photobleaching really gets to be a problem when you're doing microscopy of cell layers, and if you compute the irradiance that you have on those cells, it's much, much higher than you would ever do in vivo. But that's one of the reasons people are trying to come up with alternative exogenous fluorophores like biocompatible quantum dots, because those then aren't subject to photobleaching. Yeah.
Yeah. So when you're showing a couple of the tumors and you're showing different layers of them, have you ever constructed a 3D model, like physical model, based off those images?
Yeah, I've done a little bit of that, and other people have done more. So that's something that could be interesting to understand biology. We're talking about tumors that are so small here that typically, for skin cancers or other types of cancers, the surgeon is just carving out a big section with a margin to get it. And so oftentimes what you're looking for is just, is there any residual tumor left? Did we get it all? And so you don't necessarily, from a clinical purpose, need at this scale to do so much reconstruction. But for understanding cancer biology and how a tumor grows and metastasizes and invades the collagen, then yes, people have done that, and it is very instructive.
So when imaging cancerous tissue versus healthy tissue, are any techniques sensitive enough to kind of look at the region of interest around so that maybe information can be transmitted to surgeons as to like how big of a margin of healthy tissue or potentially healthy tissue to remove? Because I've seen a little work where they've been investigating regions around cancers, and like if you look at what depth is taken, then comparing that to cancer recurrence and stuff like that.
Yes. And that's one of the reasons why when surgeons remove a cancer, they always get this margin, they call it, because you would go, why would you remove healthy tissue? Well, it's because with some types of cancers, like glioblastoma is a great example, you don't have a clear demarcation between the tumor and healthy. You start getting this region of like abnormal infiltrate. And so you can see that if you use some targeted fluorophores, can show you that kind of the weakly fluoresce and that margin that you should take out too. Other tumors really do. I mean, it's either it's cancer cells and they're growing, and then it's a normal cell right next door. And in that case, you could actually reduce that margin if you could clearly distinguish between tumor and normal. And some of these techniques have the ability to do that. Multiphoton microscopy would be a good example of that. Yeah.
So you mentioned that the FDA does not approve specific fluorophores yet. But is there much hope for non-toxic specific fluorophores or elsewhere in the world?
Yeah, so there are some photodynamic therapy agents that are approved in Europe that could be used as imaging agents as well. That one I showed you at the very, very beginning, showing how the metastasized ovarian cancer was lighting up green. That is a combination of an FDA-approved dye and a vitamin. So that hopefully will be FDA approved because each of the two pieces are. It's just that nobody's gone through and done the paperwork for the two put together. And that could have a potential to be really powerful. Yeah.
Like how long, for the OCT, how long does it take to create the images, like the three-dimensional images of the tumor after you take the measurements?
Yeah, so what I showed you was just a fly-through of images. So there was no actual reconstruction there. That was just, looking through a 3D matrix. And OCT, they're up to now getting 1 million columns per second. So you just divide that by how many columns you want in an image and how many images you want in a stack. But there are people getting real-time volumes of tissue, which is probably faster than any physician could use it. So we're definitely to the point where you get real-time images and you can go back and forth through a volume as fast as any person's brain would want to. Yeah.
Okay, well, let's thank Jen.